Hello folks, and welcome to part 2 or objective 2 of our nervous system unit. In this part we're going to take a look at nerve cells specifically, a little bit at their structure, some of the basic parts of them, and their basic function. We'll get into the specific, how they transmit electrical signals and all that kind of stuff in a different section. We're just going to kind of give the overview at this point. All right, so some of the histology of it, you've already learned some of this before, but there's two principal cell types we find in nervous tissue. One is neurons. That's the main one that we think of, the nerve cell that sends a signal. But we also have supporting cells, glial cells, that wrap around the neurons and help to protect them and also improve their functionality in some situations. All right, so supporting cells in neuroglia. The supporting cells, neuroglial cells or glial cells, they support them. They promote health and growth. They never lose their ability to divide, which is pretty important because if you're looking particularly in the brain, where we have a lot of glial cells, oligodendrocytes, since they never lose their ability to divide, and they can divide fairly rapidly compared to nervous tissue, at least, they form up the majority of brain tumors are glial cell tumors or gliomas. So one interesting fact there. All right, move myself out of the way. So here we can see, this would be the yellow, would be the nerve cell branching off, and then we got a glial cell here surrounding capillaries and nerves and helping to hold them in place and maintain the structure of the tissue, particularly in something like the brain, right there. There are two main types of glial cells that we're going to focus on: oligodendrocytes, which is a fun name to say if you think about it, oligodendrocytes. These are branched, like the one I showed you in the last picture, and they wrap around the central nervous system fibers. You find this in the brain and the spinal column. And Swan cells, or neurolemocytes, surround the fibers of the peripheral nervous system. So you're going to see these in the arms and legs and the body cavities and things like that. This is what kind of makes, when we're dissecting the cats, this is what you'll see when you do find a nerve cell. It'll kind of be, when you can see it, it'll be kind of a white tube. And the reason you see it like that is because of the Swan cells. So here we can see, here's an oligodendrocyte. We got the cell body here, and then it branches and holds, in this case, 3 different neurons in place. Down here, we see the Swan cells, and there's a whole bunch of them along the axon, or the nerve fiber itself, with gaps in between, which we'll talk about in a second. All right, so neurons, nerve cells, they're the structural units of the nervous system. They're the main functional and structural unit of it. They have 3 main parts: the body, the axon, and the dendrites. They're relatively long-lived and have a very high metabolic rate. You use a lot of your energy, even just sitting around, to power your nervous system. Because in order for it to generate electricity, kind of like we saw in the muscular unit, you're going to be pumping ions across the cell membrane constantly. And that those sodium-potassium pumps and ion pumps, they take a lot of energy to do that all the time because your nerves are always working. So a pretty large proportion of your energy is going to your nervous system at any given time. I think it's around 10%, even when you're just sitting there and not really doing much. It might even be more because your other things aren't using energy so much. The plasma membrane is the really interesting part of nerve cells. This is the part that actually does the electrical signaling, and it's the part that does the cell-to-cell signaling. Because we saw in the muscular unit, nerves don't actually attach to muscles, and nerves don't actually attach to other nerves end to end to transmit electrical signal. There's a gap in between, that little synapse, right? And you got to get something across that synapse called the neurotransmitter to get the signal from one nerve to the next nerve. So here we can see a nice diagram of nerve. On this end here, we've got the dendrites. Lots of them. They're kind of tapering, branched. This is bringing information in to the soma, or cell body, which contains all the normal metabolic structures like mitochondria and endoplasmic reticulum and Golgi apparatus and the nucleus. All that kind of stuff is there because it's a living cell. We have the axon hillock down here, this little cone-shaped area where the axon, usually just one, carries signal out of the nerve, okay, until it gets to the end where it branches. We saw this branching in the muscular unit when we looked at motor units, right? This would be a motor unit if it was attached to muscles, 1, 2, 3, 4, 5 muscle fibers. And then we can see the Swan cells here insulating this axon with some gaps in between, which we call the nodes of Ranvier. We have a little bit later. in the notes. So the soma itself, already said, contains the nucleus and the nucleolus. It's the major biosynthetic center of the cell. It's where the neuronal processes, the dendrites and the axon grow out of it. And like I said before, it has that axon hillock as well. So we can see the soma or body here, and the processes would be the dendrites up here and the axon down here. Dendrites up on this end here, like I said, short, tapering, they branch a lot. They're generally, they are the receptive or input regions of the nerve. So the signal runs through the nerve in one direction. Here, you don't have bidirectional signaling. It's from this end down to the axon end. The electrical signals are conveyed as graded potentials. Now, potentials is what we call the voltage or the current moving through the cell. I think the proper term would be voltage. But so that's what a potential is. And a graded potential is a potential, you can think of it like a dial on a volume dial. Not when you've got, like a phone where you hit the button to move up the volume, but where you actually have a dial that you can turn it, and you can go from 0 volume all the way up to full volume, and anywhere in between. You can tweak that dial and just nudge it just a little bit, so you can get a little bit more, a little bit more, a little bit more. Well, that's what a graded potential is. In the dendrites, you can send just a little bit of signal through the dendrites, or a little bit more, or a little bit more, or quite a bit, or a full signal through the dendrites, and it will travel through the dendrites to the cell body. And that's an important feature of dendrites, the fact that they have this graded potential, this ability to send a little signal, a medium amount of signal, or a lot, or anywhere in between. Okay, it's analog as opposed to digital. It's one of the few things in the body that is analog, come to think of it. Most of it seems to be more digital, like on and off type things. So remember, dendrites, graded potentials, lots of variations in the amount of signal being sent. All right, axons, on the other hand, there's generally one arising from the axon hillock and moving all the way down here. It'll branch at the end here often, not always, but often. The terminal are these end pieces. We talked about that in the muscular system. And they generate and transmit action potentials. Action potentials, we have mentioned that before in the muscular system, didn't spend a lot of time on it. An action potential is different than a graded potential. They're both ways of measuring the amount of voltage going through the structure. Graded, right, you can do a little bit or a lot or in between somewhere. Action potentials, you're either on or you're off, right? It's either full signal, everything you can send, or no signal. There's nothing in between. It's you either got enough of a stimulus or whatever to generate an action potential and send it down that axon, or you don't and nothing gets sent. This is that binary, the on or off I was talking about before. Very important. So axons send action potentials. They either send a full signal or they don't send any signal. Dendrites send a graded potential. We'll talk about that when we talk about how a little bit more with how the nervous system makes decisions, so to speak. Okay, and at the end of the axon terminals, you secrete those neurotransmitters. Like in the muscular system, it was acetylcholine. Would travel across the synaptic gap and get the next neuron to, or in that case, the muscle to generate electrical signal and start sending down an action potential and get the muscle to contract. In the nervous system, you can have acetylcholine. You can also have other ones you might be familiar with would be like serotonin or dopamine, are other neurotransmitters that transmit signals between one nerve and the next. So let's talk about the myelin sheath next. Remember, it's sitting around the axon here, this whitish, fatty, segmented sheath around most long axons in the peripheral nervous system. And it functions to protect the axon, insulates the fibers from one another so you don't get across signals so much. All right. Not quite in the sense you'd think about, cross signals and copper wiring, but it does insulate them and keep them from interfering with each other. And the thing we're going to focus on is it increases the speed of nerve impulse transmission. Critically important. You can tell a lot about the function of a particular nerve by whether it's myelinated or not. And we'll talk about how it's able to do that when we talk about how nerves send signal in the first place. That'll be in a later one, but it does increase the speed of signal transmission. So myelin sheath is formed by Swan cells in the peripheral nervous system. The Swan cells kind of surrounds it, enclosing the axon within its plasma membrane, which is where you get the fatty, it's lipids, right, phospholipids. And it has concentric layers or rings that make up that myelin sheath. As we see here, when it's getting formed, you got the cell. It's kind of like you're pushing like a stick into a big old puffy marshmallow. But then that marshmallow keeps growing around and around and around, creating a spiral with lots and lots of layers of cell membrane. Not a lot of cytoplasm, but a lot of cell membrane there, which is what's going to do the insulation. I like to think of it as like a Swiss cake roll, right? You got all these concentric rings and circles of that white, fatty, yummy frosting in there that are surrounding it and protecting it. All right. Here's one, the nodes of Ranvier, or the nodes of Ranvier. I think they're pronounced Ranvier. I like Ranvier. Sounds more like something out of a Tolkien novel, right? The riders of Rohirrim are off to the nodes of Ranvier to fight the orcs or whatever. But these nodes, these are the gaps, those spaces where the myelin sheath, the two Swan cells, don't cover up our green nerve cell here, right? That little gap between the myelin sheath, the Swan cells, that's the node of Ranvier. Okay, that gap is what allows them to function in making the signal transmit faster. It's the gap that lets the signal transmit faster. We'll talk about that a little bit later, but just kind of giving you the structures, and then we can put it together when we talk about how the signal gets transmitted. The axons of the central nervous system. Peripheral nervous system are just about all myelinated. In the central nervous system, the brain and the spinal cord, some are myelinated, some are unmyelinated. Okay, and in the central nervous system, remember, they're formed by oligodendrocytes, not Swan cells. Those ones that are branching can grab onto multiple nerves at once. When we're looking at the brain, for instance, here's a cross section of the brain down here. We can see white matter in the middle of the brain, and around the outer edge, it's a little darker. We call it gray matter. I'm not sure how well this is going to show up. I might try to find a picture and put it in, but you might be able to see that on this as well. On the outside edge, we got the darker color, and towards the middle of the brain, we've got the whiter areas. The whiter areas in the middle are white matter. They are myelinated. The areas around the outside edge are unmyelinated. Now, I want you to think, what would be a functional difference between areas that have white matter or myelinated and areas that don't? Okay, so those areas that are myelinated, myelination is there for protection, but you don't have the protection on the gray matter, so obviously they don't need it there. So what's the function of the white matter, the myelin sheath, that would make a difference functionally? It's the fact that it increases the speed of transmission. So areas in the middle that have that white matter, they're there to be the high-speed transmission lines to connect different parts of the brain. So they carry a signal from here to here. Gray matter, because those myelin sheaths take up a lot of space, right? So if you have very small axons and dendrites, where the nerves are very close together and don't have very far to go to signal each other, you don't need a myelin sheath. In fact, it would, it's got diminishing returns. It may make it a little bit faster, but it also spreads it out, so it has to be faster. So if you got the nerves really short and really close, the myelin sheath really doesn't do any good as far as speed goes. So those areas that are gray matter, that's lots of nerves, close packed, lots of synapses, lots of computing power in there. So those would be like the computers in a big computer network. The gray matter would be the computers everywhere, and the white matter would be the fiber optic cables connecting all those computers. So they would all work together and talk to each other to get coordinated action. Is kind of the difference there in functionality. There's a couple different ways we can classify neurons. You can do it structurally. We're not going to worry about that. I'll just put it here. We're not going to test you on this. I put it in here just for interest's sake. You have multipolar with lots of processes, bipolar with just two, an axon and a dendrite, or unipolar where it's got a cell body on one end and just a long process off the other. Here's some pictures of them, just to give you some idea on what they might look like and some of the variety out there. Okay. What I'm more worried about is what we've already talked about, the functional differences. So you have ones that are sensory or afferent fibers, and they transmit signal towards the central nervous system. They're bringing in signal from outside and inside the body and shunting it towards the brain and spinal cord so it can decide what it wants to do. There's the motor or efferent neurons, carry impulses from the central nervous system out to your external body, your peripheral body, to effect a change. These are the effectors, right? And then there's interneurons associated with the central nervous system, and these are the ones that are kind of doing the decision making. Okay, back and forth between different neurons to decide on what action to take. It's the integration centers. Here we can see kind of a drawing of that. We got a receptor here, sensory neurons bringing the information in. In this case, we're showing a reflex arc. So we got the spinal cord here, and the gray matter here is in the middle. We got an interneuron, which is making the decision. We'll talk about how that works later. And then here is our motor neuron, or efferent fiber, carrying the signal out. And that'll do it for this section of the notes, guys. Join me next time when we talk about how that nerve impulse is actually sent. Check you later. Bye.